Hello, everyone. Welcome to the gene expression analysis webinar. Conduct I'll say collaboration between Azure System and Tengdahang 集团 Unimed. So today we will talk about absolute and relative quantification with standard curve method. So before we start, let me introduce Azure Biosystems to you. We are a United States company based in California. We're a team of highly experienced scientists, engineers, and entrepreneurs dedicated to accelerating your science. So what we do is we innovate in unexpected places. We create products that combine smart and simple workflow, thus giving you the raw data that you can rely on using the simple workflows that you can trust. Azure System is founded by Arnold Shifji, our CEO and chairman, and Dr. Diping Che. So Dr. Diping Che is our executive VP of R&D. If you're looking through his CV or his past experience, he has a lot of experience in terms of developing imaging system for protein expression from Illumina and Alpha Innotech. So as a fact, Azure System founded in 2013. Most of our core members are coming from Alpha Innotech, which is now the brand will be called as Protein Simple. So at the moment, Azure System, we have a few key instruments over here. The first thing is we have Sapphire Biomolecular Imager, which is the industry's first hybrid CCD imager and laser sequencing system. And another one is Azure Imaging System, which we focus on Western blot imaging. And today, our highlight will be on Cielo Real-Time PCR system, which we launched last year to enter into the genomic market. Right. So here's our agenda for today's session. We will talk about what are the quantification strategies in analyzing the real-time PCR data or qPCR data. Most of them today, I will only talk about absolute quantification with standard curve and also relative quantification using the standard curve as well. So we will talk about the principles, the data, how you normally do the analysis in a general touch, and also some publication example on how people advance this kind of analysis. Right, let's jump into the first agenda, which is what is the quantification strategies in real-time PCR. In general, there are two strategies can be used in qPCR. First, absolute quantification. Second, relative quantification. What you do in absolute quantification is it relates the qPCR signal to an input copy number using a calibration curve or what we call as a standard curve. So this is pretty much straightforward. You get your data from the standard curve. That's it, nothing else. For relative quantification, by the end of the day, you want to measure the relative changes in gene expression levels. You can use them by the means of the standard curve or without the standard curve. Right. Here's a simplified or say figure kind of summarizing what are the quantification strategies in real-time QRT-PCR. So I think some of you might be very familiar with this chart or this figure where you can find on the website and from this author that is very well known in qPCR knowledge community. So here we have absolute quantification and relative quantification. So let us look at the first one, absolute quantification. As you can see, in absolute quantification, it is a must to have an external standard curve. Either you're using one color detection system, which is using just a cyber green, or using multiplexing detection system, like you're having different probe dyes by the end of the day, you still need to establish that external standard curve. Okay, so in order to establish the external standard curve, so you get the data for your unknown sample, you need to pick what kind of sample or what kind of the standard type that you want to use to establish or generate the standard curve. So there's a lot of them. You can see the list of them coming here. RT-PCR product of your gene of interest, plasmid DNA, in vitro transcribed RNA, and some commercially available synthetic DNA or RNA oligos. So absolute quantification is very, very straightforward. Let us look at relative quantification. Relative quantification here, as you can see, it comes down to two big branches. By all means, you definitely need to do normalization within the sample itself. So this is not the same as the absolute quantification. In relative quantification, you will need to do normalization either by using one reference gene or what we call as housekeeping gene, or more than one, or maybe up to 3, depending on your expression level. And also, if you need to do, you also have the option to do a standard curve in the relative quantification. This method is what we're going to discuss later, which is called relative standard curve quantification. So as you can see, under the relative quantification, we have the very, very famous delta-delta CT type of analysis, which is without doing the real-time PCR efficiency correction. Like with this part, we will talk about it later. And another one, which is what we're going to talk about today, which is the relative standard curve quantification, will be using real-time PCR efficiency correction. Then you can use all other third-party software or any other software that comes with your qPCR platform. All right, so the reliability of an absolute quantification real-time PCR assay depends on the condition. It will be very reliable if both unknown sample and standard, the target of interest, okay, target gene, have the same amplification efficiency. So this is very true. We're talking about qPCR efficiency, what it is. So then if you have a certain knowledge of PCR or qPCR, you know that the underlying, also principles of PCR is you expect twofold differences, or not say twofold, 2 to the power of 2 differences. I'll say double up the differences every single cycle. So you have double every single cycle. So that is what we expect. So we get the amplification efficiency of 100% means by the 2. And on the other hand, when you're doing relative quantification, it's much more easier to perform than absolute quantification, because you don't really need a standard curve. It is based on the expression level of your target gene versus the housekeeping gene here. So that's why the normalization. And then you compare between untreated group, untreated sample, control group, or maybe a blank, and so on and so forth. In theory, this is enough to cover the purpose of doing relative quantification, because you want to investigate the changes in gene expression level, so you do not really need an absolute value. The units are used to express the relative quantities are completely irrelevant as long as both them on the sample or the unknown are the same. Thus, you can use these across many different experiments. Right, let's look at one by one. First, we touch on the absolute quantification with standard curve. So today we will all talking about standard curve today. Right, what is standard curve? Standard curve allows generation of sensitive, specific, and reproducible data, because you have something to rely on that you can refer every single time. Or if you do it on every single assay, on the plate itself, every single time you test it on your target unknown samples on target gene, you generate the standard curve every single run. This allows you to have the control that you're confident with your assay every single time. Right, absolute quantification with standard curve method assume that both target gene and the standard having the same amplification efficiency. So target here also means by your unknown sample. How to generate this standard curve is you use a dilution series of nucleic acid template by your choice with a known concentration. You know how much or how many copies or how many grams of tissue generated for this nucleic acid template to actually give you that standard curve. You need to plot it. So by the end of the day, the example application for absolute quantification by using the standard curve is you can correlate, let's say, the viral copy number with a disease state. Sometimes if you have a higher copy number of virus, that means the disease state is much more severe than the lesser copy number of viruses in the sample itself. Here I'll be using tenfold serial dilution to tell you how the standard curve should be looks like. So the qPCR result of a tenfold serial dilution of a standard need to be plotted as a linear regression of the CQ values versus the log of the nucleic acid quantities. Therefore, you have the standard curve. I know this is a bit mouthful here. There's a lot of mathematical also equation here involved, but I will not touch on that. So we just talking about a general sense and how people use these for and improve it definitely. Right. So in the standard curve, what kind of information that you can get or you need to see or you need to check out is the slope and also R square value. So here we're using the tenfold dilution standard curve to give you what kind of slope or R square value that you should expect. Slope means the slope of the line, the linear regression itself, how slanted it is. So you will always get the slope in the equation, the linear equation that you plotted in the standard curve, from the standard curve. So in the tenfold dilution standard curve, anything between minus 3.1 and minus 3.6 is acceptable. This also means that this qPCR or PCR assay has 90 to 110% of PCR efficiency. And for tenfold dilution series standard curve, a slope of negative 3.32 means you have 100% PCR efficiency. So what does it mean? We keep talking about PCR efficiency. What is it? It means how efficient or how your PCR run is fulfilled with the expected or assumed amplification number. Let's say you have one copy number in the cycle as a starting amount, then you should expect 2 copies by the end of the first qPCR cycles, and then 4 copies by the end of the qPCR cycle 2. So this is what it means by 100% PCR efficiency. Sometimes you have lower, sometimes you have higher qPCR efficiencies, depending on, these are heavily influenced by either your sample or primer or even the reaction mixture that you use. So this is something that you need to do, I'll say troubleshoot and try before you run it on your unknown samples. Right. Slope. Next, we go to R square value. R square value is a measurement of the assay's performance. What it means is the coefficient of correlation between the generated data and the expected results under ideal condition. So above 0.985 R square value is generally acceptable. Depending on lab to lab, some of your PI may say, I want the R square value of the standard curve is above 0.995. So this is how you decide or cut off based on your own experiment. But in general, 0.985 is okay. All right. So what it means by does it mean, this value? So if you have 0.985 R square value, it means more than 98.5% of all the total variations in the nucleic acid sample used to produce this standard curve can be explained by the relationship between the CQ obtained and their respective nucleic acid. That means your linear equation from this standard curve have 98.5% to be true. That is the thing. And to be able to explain and get the result appropriately. So that's why R square is also very important to actually check out if your standard curve is running okay. Right. So in terms of the information you can get from tenfold dilution standard curve, you by the end of the day, this is how you get your nucleic acid quantity if you have an unknown sample CQ values. Like the nucleic acid quantity in the unknown sample is 10 with this kind of equation, 10 to the power of CQ value minus B divided by M. Okay, so this is the equation that you can use to get the nucleic acid quantity from the standard curve. Where B is the y-intercept, so this is what you will get from the linear equation, and M is the slope of linear regression. Right, so this quantification data is always reported relatively compared to a defined unit of interest. For example, by the end of the day, the data you get is, for example, how many picogram of tissue, how much the concentration that you get in your unknown sample, like microliters, or how many copies of gene per unknown XX nanogram of the total RNA, or how many copies per gene, and how many copies per cell, and so on and so forth. So you have a definitive value that you get from this standard curve. So this unit needs to be specified by the users. So you need to specify this unit based on how you generate the total RNA by itself. Either you get it from a tissue, either you get it from the cell count, or so on and so forth. So this will become the constant factor, and that will give you the specific units, and therefore you know how what the value should report by the end of the day. And because you need to quantitate the gene expression data from unknown sample by interpolating the CQ values into the standard curve, right? So in another word, if the dilutions that make up the standard curve is measured in picogram, for example, then the unknown sample will also be measured in picogram as well. Right, here I'll be using a data show in Azure Cielo Manager, Absolute Quantification. So we have 4 genes over here. We're doing multiplexing. You have 4 standard curve over here. You can see that in each of the target, you have the efficiency, okay, the qPCR efficiency, the R square value, the slope, and also the y-intercept. And do remember that different dilution factor will give you different type of slope, okay? But you will different kind of slope range, ideal slope range. But R square value generally is a very good idea to check on whether your standard curve is okay for this run. So let us look at the first gene. The first gene has the R square value of 0.993. So if you feel that this is fit, that means then you can stick on it using this standard curve. If it's not, the R square value is a bit too low or too high, it's not reaching up the efficiency, then you might need to tweak on your either primer or template quantity, or even your reaction mixture itself. So this is pretty straightforward. You have a serially diluted standards, okay? Either you dilute them in tenfold dilution, then when you do the analysis on the software by itself, you assign the definitive quantities that you put. Over here, you can put either using a designated unit, or like something I say, weight, copies numbers, the number of cells, those can be the quantity by itself. Once you get the standard curve, okay, from the standard itself, serially diluted, and assigned quantity, the known quantity, when you have an unknown sample, you will get the CQ value. So you won't get any starting quantity. You do not know. So from this unknown sample, you only have CQ value. So you want to know how much starting quantity on my unknown sample. So here standard curve come to play. You extrapolate this standard curve, CQ value. For example, your unknown sample is having CQ value of 30. So you plot it like a normal, how you get the value from standard curve from 30 CQ value, dot over here, then come down over here. So you have roughly, I'll say 10, maybe 12,000 or maybe 20,000 of the copy number. So if you have a qPCR analysis software, they normally will automatically analyze for you. Okay, so it will indicate for in the unknown here. So can you see this is basic or standard, so we don't see any unknown samples. But in the unknown sample, the software will automatically calculate the quantities based on the standard curve of designated target. Right. So you can see that this is everything about standard curve. So most importantly, the curve must be thoroughly validated, as the accuracy of absolute quantification depends entirely on the accuracy of the standard curve. So a bit of mouthful here, but that means that since you're using standard curve, everything is about standard curve. You need to validate the standard curve. You need to make sure the standard that you use is true to your own unknown sample. It can be extrapolated. And there's a lot of factors can be contributed to inaccurate standard curve. For example, the material of your standard, the type of your standard, and how you design the standard, maybe even the concentration you want to put in to generate the standard curve, and production of the standard curve. For example, if you want to go for commercial or you want to self-generating the standard, then you want to make the standard curve. And another thing is the stability of the standards over long storage time, since you might need to repeat the test over and over again for a longer time. And also a batch variation in reagents as well do bear that in mind. All these will contribute into an inaccurate standard curves. So accurate and reproducibility of a standard curve relies on standard type and also good lab practices. So here, good lab practices means you have proper documentation, proper pipetting skills, everything is according to the plan, proper calculation and preparation and optimization beforehand. So there's a lot of things to do here. qPCR is a very sensitive technique. Any one of a thing is not working right, your whole qPCR data will be messed up. For example, if you have contamination, then you need to eliminate the contamination. Okay? Right. Let me talk about how the standard type can influence the outcome and reproducibility of a standard curve. You can use either genomic DNA to build the standard curve, purified RT-qPCR product of your gene of interest, commercially available synthetic oligonucleotides, or you make your own recombinant plasmid DNA or recombinant RNA from your target of interest. You can do that as well. All these serve the purpose to give you that reproducible standard curve every single time, and that you know that your unknown sample have the same efficiency of your standard on your together with your standard. Right, let us look at one by one. So you have purified PCR product and some commercially available synthetic oligonucleotide as the standard type you use to generate the standard curve. These are quick, okay? These are quick, but they are not really stable because you see, you have your purified PCR product, then you use it to prepare the standard curve and you re-amplify them again. So there comes the re-amplification problem. And what if you want to do recombinant plasmid DNA? This this method is rather stable because it's a DNA. You do not worry that it might be degraded and so on and so forth, but it requires cloning and also purification, and also this is also linearization problem because plasmid are normally in circular, they are mostly circular plasmid. How about recombinant RNA if you want to use it for? So if you can also simulate the natural mRNA if your sample is actually reverse transcribed for mRNA, then this will be okay because they are going through the same process. You're doing RT-qPCR together on your unknown sample and the standard, which is recombinant RNA here. So you ensure that both of them go through the same process. So it simulates a natural mRNA in your sample, but it is not very stable, like say, because its RNA is easily to degrade, and of course, it's very easily to influence by all other factors. And this also requires a very complicated cloning and linearization problem as well. So all this information, you can look it up on the literature. So why all different types of standards will actually addresses or give you different types of disadvantages. So most importantly, you need to find the one that fits into your own sample, the unknown sample to be exact. So if you're doing genomic DNA, qPCR or genomic DNA, then by all means use genomic DNA for Pcr to be your standard. That's how it works. Right. So there's a very common practice here that you generated the standard curves, okay, but you're not running it on every single run. You basically copy and paste the previously performed standard curve. You use it to extrapolate your, let's say, second run or third run of the qPCR. So this method is very quick and easy because you're just basically using the previous standard curve. But you do remember that it does not cover the variations in qPCR experiment. Every single run is different. The pipetting might be different, even though you have the same personnel, the same person who pipetting it. Even you're talking about ambient temperature, the set of pipette that you use, even the batch-to-batch variation in reagents, all these will induce variations in the qPCR experiment. Therefore, it is I would say it's recommended to run standard curve in every single assay every time you run. Thus, we encourage you to use 96 well plate to cover your sample, unknown sample, and the standard curve for everything. Another thing is you can do the standard curve, but make sure that you have everything constant, like the same batch of reagents, the same person, the same set of pipettes, and so on and so forth. Try to control it and maybe do it within a week. So you cramp down everything within a week and do it properly so you don't need to set up or generate the standard curve every single run. So this is also one of the options, depending on your lab, how stringent and how controlled you want the data to be. Right, so here we look at the publication. Here is an example of how people try to set up or develop a MyKey compliant real-time PCR assay for this is, if I'm not mistaken, will be Aspergillus species detection. So you can see the very famous Stephen Bastien name over here. So basically what they did is they have standard curve generated 8 times, basically 8 standard curve plots using the genomic DNA. The reason why they do that is because they are doing the detection itself from the unknown sample. The unknown sample is also containing genomic DNA. Thus, they're using genomic DNA to be the standard type. You can see in the text over here, the hydrolysis probe-based assay designed to target the 18 ribosomal RNA DNA sequence of the Aspergillus species and have an efficiency of 100%. Here they claim 100% because all 8 standard curves over here having a range of 95 to 107% of efficiency, and they have covering the dynamic range of at least 6 orders of magnitudes. 6 orders means six zeros, I'll say. And they also tested the limitation of detection and quantification, so on and so forth. So this is how you develop a stable, I'll say, and reproducible qPCR analysis strategies, quantification strategies for your assays, for your experiments. So thus they say that this is robustness of specificity and sensitivity make these an ideal molecular diagnostic tools for clinical use. Right. Then next, let us look at another one. This is a very old paper, which explain a method for absolute quantification of cDNA and using real-time PCR. So here you can see they generated a standard curve using that for the total DNA quantification. They're using lambda DNA as a standard. This is a commercially available standard, and they use it in serial dilution and generate the standard curve over here. So, and this is the reason why they use lambda is because they have the unknown sample that they use is actually have doing a lot of series of cloning. So let's say here they do cloning the product of the qPCR, and then they verify it with using DNA sequencing to confirm the sequence of the clone product. Then they use the purified plasmid, cut it off, and then and plot it against the standard curve or generate the standard curve and make sure that they are having the same qPCR efficiency. Then you know that now the unknown sample can be used to get the DNA concentration using the standard curve. So this is how you make sure that both of them having the same efficiency by matching your unknown sample, what you want to detect from unknown sample, and the sample type that you use to generate the standard curve. Both of them must be matching each other, so you know that you are safe to extrapolate the data on the standard curve by itself. And next, we have extended one. They're also addressing the concern about cloning. They're basically using recombinant plasmid DNA. That's why they're using lambda DNA as the standard. So what they want to do is they want to determine if a circular DNA amplified having a different efficiency than the linearized DNA. So here, because they are using circular DNA to become the standard, or to use it to detect in the unknown sample. So, and this is they want to make sure there's no differences over here. So they do it, they cut up the plasmid DNA, and then having a linearized DNA, then to do the qPCR, establish and generate the standard curve, just to check that whether they have the same efficiency and also the slope. So you can see they're having very similar, okay, slope and efficiency. So they feel safe that they know, even though they're not using circular DNA in as the standard type, but it's okay because they're having same efficiency as the standard they use. Right. Next is an example how people enhance, I would say, advance these analysis strategies, which is they correct the quantitative PCR efficiency variation for the microbial community samples. I highly recommend you and go and read this up. They addresses the issues that were of using standard curve method in the absolute quantification, because as I say, it assumes that both the standard and sample target template having the same efficiency. So if let's say you're not using the standard that match your sample type, then you might have a problem over there. So there's a lot of equations that derive from this, and then I think you can actually look it up and see how they generate and kind of propose and validate these kind of correcting methods. All right, that's pretty much for the absolute quantification with standard curve. Next, let us look at relative quantification with standard curve. So this relative standard curve quantification, also called as E method, it can produce more accurate relative quantification data because it is based on the true PCR efficiency of both target gene and housekeeping gene. So this is much more, I'll say, reliable in this sense because you count in the PCR efficiency here. It is easier than absolute standard curve method because the quantity is expressed relative to the standard. So you have a standard. The standard can be either your untreated control, a time point zero, or even an healthy individual. So by the end of the day, you want to have a quantity that expressed relatively to your healthy people, like higher or lower, how many fold higher, how many percentage lower, that kind of sense. So any nucleic acid template containing the target gene and the housekeeping gene can be used to prepare standards by serial dilution. So you don't have restrictions over here. Anything that contains your target genes and housekeeping gene, you can use it to set up the standard curve. So since you're doing relative quantification, the standard must be accurately diluted, as their relative dilutions are very important to plot the relative standard curve and quantitate the relative expression of the unknown sample. So here will be rely a lot of, I'll say, accurate calculation, mathematical calculation, and also very good pipetting skills in diluting, doing a very good serial dilution. There's a lot of tips and tricks over there, like don't change your tips and avoid bubbles, so on and so forth. So this is very much reliable, very relies on pipetting skill. And for relative standard curve, it uses a set of relative standards with unknown sample. So you don't need to have a known sample for this case. You just having unknown sample is fine as long as you do the dilution properly. Okay, right. So now the standard that you use will be the 100% or the one time or the control, where the other unknown, okay, the unknown sample will be expressed as n-fold differences relative to the standard. For example, for for example is my or say transgenic cell line having twofold higher gene expression compared to the wild type, or let's say, yeah, or wild type cell line. So this is how it works. You need to relatively tell the differences. So but in that sense, because this is a relative quantification, you still need to normalize within yourself, within the sample itself. So you need to prepare standard curve for both target gene and housekeeping gene in order to do the normalization. The amount of target and housekeeping gene will be determined from their respective standard curve. Thus, the choice of housekeeping genes or lineage specific genes are very important. So, what kind of housekeeping gene that you want to pick to become the denominator to normalize sample to sample variation? So, this is very important. We recommend you do more than one housekeeping gene. If you're doing multiple pairwise correlation analysis to get a summary reference gene index. Here's an example, a very simplified diagram that I set up for you to get you a concept of how it looks like in relative standard curve quantification. So here you have a serially diluted standard. This standard can be anything, any nucleic acid that have target gene and housekeeping gene, a proper expression of it. So let's say in my case, in my studies, this will be my the pool sample from all my from the, let's say if I'm doing brain study, it will be a whole brain RNA. That is the case, and of course a cDNA in this case, right? Then with that, after you do the reverse transcription, you get a cDNA, then you dilute them in 1 in 20 to get rid of the salt and buffer and so on. And then you do the qPCR run with the serially diluted standard, and you get on the standard curve for both the target gene and your housekeeping gene. So I would be using one housekeeping gene over here just to simplify the explanation. So this is the standard curve part. The part for your unknown sample, you need to do in the same dilution as the standard, the one that you I'll say the crude sample, before the serial dilution. So here will be in the same dilution here, 1 in 20. Then after that, you do qPCR, getting the CQ value of your target gene and the CQ value of your housekeeping gene. From this, you plot and get the X unit, okay, from the standard curve generated from the standard. Okay, same thing goes for the housekeeping gene. And then once you get the unit, you normalize it. What you get is you normalize the target gene, okay, divide the X to Y, all right, then you get the normalized target gene in 1 in 20 diluted unknown sample. You need to pair them together. Your standard is 1 in 20 diluted. Your sample also need to be 1 in 20 diluted, okay? So they are on the same ground. So you compare apple to apple. You are not compare small apple and big apple. That's the sense. So in order to get the relative differences, you're dividing the target. So sorry, I think this is a difference. Should be B divided by A. There's a typo here. So then you get that the target gene is how many fold difference they're relative to the standard over here. So this is what we get. The standard here can be your wild type, like I say, can be your unknown, can be your total protein or anything, total RNA or anything. So as long as you have the standard, that you know that you're getting everything is fine. The qPCR efficiency is correct, then it's fine. So over here, why it says is count in the qPCR efficiency, because they are using the same primer set. Remember here, you have the same primer set for your target gene that will amplify in both the standard and also your unknown sample. So here they count in the qPCR efficiency by the means that making the primer set constant, and also everything is standardized. You have the standardized diluted in the diluted standard, and standardized diluted unknown sample. Right, so this is what you can see from relative standard curve of using sort of Cielo Manager. You can see here we have basically 4 sets of sample over here, and each of them actually targeting GapDH and CMYC. Okay, we have the unknown sample, all right, and also the sample number 2. So when we have a sample dedicated for a GapDH and also CMYC. So by the end of the day, you will get 2 standard curves over here. So this standard curve will apply for the unknown sample to plot the CQ value and get the starting quantity that you will get later. Right. So if you, like say, certain third-party software or the analysis software that comes with your qPCR platform will automatically calculate the quantities for you based on the standard curve. So, and by the meantime, you also get, they also compare between the group once you set up the reference sample, then you will get this comparison, how many fold differences are lesser than your control sample. So let's say here the control sample is sample 1. This is how it works. So this is what we call as relative standard curve method. Right. So there's a lot of people or a lot of researchers actually publishing here. I know this is a comparison or methods for relative quantification use of gene expression using real-time PCR. This is an example how they do the relative standard curve method. You have standard curve for efficiency, qPCR efficiency calculating the target, the nucleic acid, and also the endogenous reference here will be the housekeeping gene. So you can see both having a very good linear R-square value, and the slope is okay as well. Yeah. Right, so this method based on the qPCR included the standard curve method, the efficiency calibrated method, and also the very famous delta CT or CQ method. They analyze the 3 methods together and tell us that the efficiency calibrated standard curve are the most stable one for quantifying small differences in relative gene expression than the delta CQ method. Okay, so I will highly recommend you and look it up on this paper and why they come up with such conclusion. But do remember that there may be differences in terms of the gene that you want to target. It may be applied to the gene of interest, but it may not apply in your own experiment. You need to look it up. I'll say the best thing is look it up on your literature on how people do the analysis, and then see whether it fits in your experiment. Then you can adopt it. Right. Another thing is addressing the correction for efficiency is very important. So you can accurately report and quantitate and analyze your qPCR data. So here we say that you have a standard curve plotted based on the dilution series, and then plot the standard curve. It looks nice, all right. But when they actually plot the actual dilution, how they get it is they get it from the amplification curve itself instead of the dilution series that you assume, because of all this is due to the, I would say, human pipetting error and even qPCR efficiency of the machine and reaction meter itself. Right. So what they do realize is the differences in these intercepts do reflect that you actually do have different amount material in the undiluted reaction for each of the series. I will highly recommend you also look at this to actually see why they go out and tested this, because I believe that they do see inconsistent data, I would say. Thus they kind of propose this kind of mathematical strategy and to correct the standard curve itself. Right. So let me introduce Azure Cielo Real-Time PCR system to you. We come to the part that I will introduce the qPCR system. This is a standalone machine, an open platform supporting both dye and probe-based chemistry in the qPCR reaction. So in terms of performance, it has unique optical design that delivers both sensitivity in reproducible data. You can put there's a lot of dye choices that is compatible with this machine. We have touch screen based in workflow. Say very easy for you to set up your experiment. You can connect it via Ethernet, LAN, Wi-Fi, and USB. So the innovative, I'll say, high performance optical design over here is basically two sets of 16 fiber optics. One set coming from the high powered channel specific LED, so give you the very specific excitation for each of the dyes. Then you have a detector camera with a filter that's specifically for that dye channel as well. So therefore you have total, I'll say, a whole well or total well detection over here. And then so you won't miss out any single signals. There won't be signals that you didn't capture. And you capture them, therefore you have higher sensitivity in this sense. The pixel will be captured more and to get the data. And one keynote here is this design avoids the need for passive reference dyes such as rocks, because you do not need to normalize the differences between the optical path itself. So yeah, the data is what you get. What you get from the reaction, that's what you get from the machine the machine will capture. Azure Cielo comes with two configurations, Cielo 3 and Cielo 6. Cielo 3 will have 3 channels available, and Cielo 6 will have all 6 channels available for you. So it can do up to 6 multiplexing, okay, and without any crosstalk at all. You can also do custom dye and probes, that is, as long as the excitation and emission wavelengths are compatible with the dye channels. It's capable to do interval uniformity detection in the machine, accurate temperature, ability to detect twofold differences in gene expression, multiplexing without or low signal crosstalk, bell curve analysis, reproducible from run to run up to 1,000 continuous run, and also broad application compatibility. This machine is very durable, and also the reason is there's no I'll say 1.5-fold differences is because that is a limitation of a qPCR machine. So that is a technological based limitation here. Right. So Azure Cielo, there's a list of selected customers. We have a lot of users all over the globe. We have Perkin Elmer and institution, university, and even diagnostic labs and developers in terms of the kits and everything. Therapeutic kits are using Cielo for their validations and even testing. In Asia Pacific region, we have came in from Singapore and Sariroti from Indonesia, and for Taiwan will be China Medical University. So we have customer reviews here actually trying to tell us that how Cielo are capable of solving their difficult questions. For example, if you have very low quantity amount of nucleic acid template that you cannot longer max it out, and your target gene is also lowly expressed, then you might need some qPCR machine that is capable to detect the small differences and also very sensitive to all the signals that you get. Therefore, the optical design is very important over here. It tells you what you can get, and it will, if your experiment is okay, like having good expression, that is fine. But if you encounter something like very lowly expressed gene, but you cannot increase the RNA load anymore, then I think you might need to try or have the option to try different kind of qPCR platform to see whether it gives you that the data that you want. In this, why I'm putting this out, because Unimed Teng Dahang Jituan, okay, sorry, my Chinese is not very accurate here. They have a demo unit of Cielo 6. I would highly recommend you, if you're interested, go and request a demo or even a trial run, so you see whether you like the machine or not. All right, so this is the summary or the brochure that you can that summarizes what Azure Cielo capable of doing it, and then the sensitivity. So we have also 3 application notes available on the website, or if you're interested, you can request from Unimed as well. We're talking about why this machine can do 6-channel multiplexing, and why we can push the limit to detect sensitive and accurate single copy amplification, and why we say this optical design eliminates the need for passive reference dye. There's a lot of information that you may not be able to get out there that in the publication that will not tell you in terms of the technology in these instruments. So let us look at what are the agendas, or we'll say in recap what we talk about today. So we talk about quantification strategies in real-time PCR. Basically, we talk about everything about standard curve, absolute quantification, and relative quantification with standard curve. The principles are the same. You want to generate the standard curve. So the standard itself is very important, particularly with absolute quantification. With relative quantification, the standard that you use can be anything as long as it has, but you can use unknown sample as well. So basically, they are mostly unknown sample, and you just need to do the dilution accurately. That's it. And we have talked about several publications and how people improve on these strategies. Right, thank you everyone. I will open up the floor for Q&A session. Please feel free to voice out your question, and so we can address your question here. Thank you.
大家好，刚刚的线上讲座，大家对于刚刚的内容有没有什么样的疑问或者是问题呢？都可以用中文发问或者是英文发问都可以，或者是你可以直接打在那个chat box 上面，如果大家目前没有问题的话，那如果之后有想到的话，也可以直接用信件或者是用电话直接跟我们来联系都可以。好，那这边我们有一个feedback form， 请大家就是在那个 chat box 上面有一个连结，对，那请大家可能花个一两分钟的时间帮我们填写一下 feedback form， 就是您对于今天的讲座的内容的一些 feedback 可以提供给我们知道，还有就是如果之后有没有我们要再办线上讲座，你有没有想要听的 topic 也可以告诉我们。
All right. Thank you everyone for coming. 非常感谢大家的参与。然后请填写这个讲座回馈表 webinar feedback form. So we know that what we need to draft the content next, so you can know better in terms of the qPCR knowledge, or just tell us what you want to hear. Okay, so we are Azure Biosystems. So if you have any inquiries or any application question, please feel free to email me. And we also have Nicole here with us today. She's a regional business manager. And then if you have any inquiries, also you can also direct to Priscilla. We're here with the contact information over here. Okay, thank you everyone for coming for this webinar. I'll see you guys next time.
好，谢谢大家今天的参与，然后我们会在近期内就把这个饮料券分送寄给那个前3名的参加者，谢谢大家，
谢谢 ，Thank you everyone，
拜拜，
拜，谢谢大家。小文这个 recording 会在
谢
哪里可以看得到？
好的，
好 ，Thank you Nicole，Thank you everyone，Bye bye，Stay
谢谢
safe。
大家。
降2级咯。
降2级了
之后我们
哦，降2级了。
今天的讲座录影档也会放在我们的官网上面，大家有时间或是想要再温习的，都可以在我们的官网上面去观看。
好的，谢谢 Priscilla，
谢谢大家。